Hi everyone. Okay, so on to part 2 of the lecture. So in the second part, we're going to look at the applications of ocean colour data in the context of the ocean carbon cycle. So we're going to go firstly through what the Earth's carbon cycle and then the ocean carbon cycle, and then how ocean carbon satellite products are used to understand carbon cycling in the ocean. So we're going to go through a whole range of satellite, primarily ocean colour based products that can be produced related to the ocean carbon cycle. And then we'll finish off by talking a little bit about how these observations are now being integrated with robotic platforms and with models to look at the pools and fluxes of carbon that we can't see from satellite. Okay, so a bit of a background to this. On the left here we have the global carbon budget. This was from their 2019 paper. So this global carbon project basically provides a budget of carbon throughout the Earth system every year. Now, these numbers here represent the flows of carbon between the ocean atmosphere and land. So in terms of anthropogenic emissions of carbon, so we have around 11 gigatons of carbon being put into the atmosphere from the burning of fossil fuels and land use change. Now around half of that stays in the atmosphere, and the remaining half is either absorbed by the land or the ocean. So the ocean plays a very important role in modulating the atmospheric CO2 concentrations. Now, as shown on the right image here, we have the increasing atmospheric CO2 concentrations at the Mauna Loa Observatory in Hawaii. And we can see that the atmospheric CO2 concentrations have been steadily increasing since since the Industrial Revolution, but certainly on this time series since the 1960s, and we're now up beyond 400 parts per million at the moment. So atmospheric CO2 is increasing. That's having a profound impact on our climate, which I'll talk about in a bit. But in the context of the second part of the lecture, the application we're looking at is using ocean colour observations to understand the cycling of carbon in the ocean. Now, this is a schematic diagram of the ocean biological carbon pump. It's quite rich, a lot of information there. But if we just run through some of the key components of this, we have 4 different pools of carbon in the ocean. There's dissolved inorganic carbon, which is the largest pool of carbon, DIC. There's dissolved organic carbon, shown in the green here, which is, again, a large pool of carbon in the ocean, around 662 petagrams of carbon in the ocean. Then we have particulate organic carbon. Now, particulate organic carbon includes algal and non-algal organisms and other organisms and particles in the ocean, including principally phytoplankton, zooplankton, bacteria, and detrital pools. Now, this is a small pool of carbon, but it's the most dynamic. So the phytoplankton pool particularly is constantly turning over, which ultimately contributes to the sequestration of some of the carbon in the deep sea. And we also have our particulate inorganic carbon pool, which is a small pool principally related to the calcium carbonate cycle. Now, the satellites, the ships, all of the tools we use are illustrated in this figure to monitor these different pools, but satellite plays a very important role. Now, how carbon then flows between these different pools depends on the processes here, which is shown in the bottom right here. And I'm not going to go over in detail this figure, but as you see, there is a conversion between dissolved inorganic carbon into particulate organic carbon through the processes of primary production, and that drives a series of other processes in the surface layer of the ocean. Now, this carbon, principally produced through primary production, can then be transferred to depth using various pathways: physical, the sinking of dead phytoplankton, the sinking of aggregates, and also this process known as zooplankton migration, where zooplankton eat the phytoplankton and then migrate to depth and poo out or in fecal pellets, or does release the carbon that they've eaten. And a small portion of that carbon can then flow down into the deep sea. Okay, so we can use satellite observations to monitor some of these pools and these processes in the ocean, as shown by the red boxes here. Now, just to put this into a bit of context, how important the ocean biological carbon pump is. So this is this process where phytoplankton produce carbon, or convert DIC to organic carbon, and then some of that carbon can sink through the ocean and be locked away at depth. So it's believed that the ocean biological carbon pump exports between 9 and 12 gigatons of carbon per year, as estimated from this LAWS study shown there. Now, they produce around 50 gigatons phytoplankton. Around 10 gigatons is exported to the deep ocean, the ocean interior. But then only a fraction of that actually makes it all the way down to the bottom of the ocean. And again, only a fraction of that is actually buried in the sediments. So it is a small component that's actually buried in the sediments, but over long time scales, it is critically important. And that's illustrated by this statement here, where a study showed that if the ocean biological carbon pump were to be switched off, it's estimated that atmospheric CO2 concentrations would be 50% higher than they are today. Okay. Now, the ocean carbon cycle and the ocean biological carbon pump is also at the core of a recently funded European Space Agency project called BICEP. So it's Biological Pump and Carbon Exchange Processes. And I'm a participant of the consortia involved in this project. And our goals within this project is really to bring satellite observations into the limelight of ocean carbon research. So we have a series of tasks and activities building on previous ESA activities and other international activities and recommendations, where we are going to be producing a series of ocean carbon products that can ultimately help us paint a better picture of the ocean carbon cycle. Okay, so let's start running through this. This is another schematic diagram of the ocean carbon cycle. Our different pools of carbon in the boxes there. Now, the green parts of this image indicate those components of the pools that we can access from satellite observations, or that satellite observations have been used to try to look at these pools. And the red ones are kind of the areas that we can't use satellite observations through. Now, we talked about the chlorophyll concentration briefly in the previous lecture, so I'm going to jump straight in at monitoring total phytoplankton carbon from satellite observations here. So we know we can look at the chlorophyll concentration, as it has this imprint on the ocean colour signal related to the unique absorption properties of the chlorophyll A pigment. But there's also been techniques that have looked at estimating phytoplankton carbon. So this is considering carbon as the unit of biomass rather than chlorophyll. And there are certain advantages of doing that. Certainly, ecosystem modelers particularly like to work in carbon units. So these are just two techniques shown on the left here. One technique actually relates the phytoplankton carbon to the chlorophyll concentration, shown at the top there in the red line. And the other technique actually that's been used now by various scientists is to use the backscattering signal as observed from satellites and relate that to the carbon in the phytoplankton. Now, if we know the chlorophyll concentration and we know the carbon concentration from satellite, we can actually produce maps of the carbon to chlorophyll ratio. Now, this is an important ratio in marine ecology and biogeochemistry, because it tells us something about the physiological state of the phytoplankton, so how they've adapted to the light environment. So you can see a much higher carbon to chlorophyll ratio in the oligotrophic gyres here than you can, say, in the polar regions, and it basically sort of illustrates how the phytoplankton have adapted their cellular machinery to be well suited to a hot highlight environment in the oligotrophic gyres, and also change their cellular machinery to harness the environmental conditions in the polar regions as well. Now, validation of phytoplankton carbon products is an ongoing exercise. It's actually a very hard thing to measure using in situ observations, the phytoplankton carbon content. And this is because there's carbon in many other different particles in the ocean, and it's sometimes very hard to separate out the phytoplankton and the other components. Slightly easier with chlorophyll, because chlorophyll is unique to phytoplankton. However, validation exercises are ongoing and indicate some reasonable performance in the phytoplankton carbon products. Okay, so next I'm going to talk about particulate organic carbon. So this is the carbon in all particles in the ocean. It's a slightly easier thing to measure, I would say, than phytoplankton carbon. And various techniques again have been developed to estimate POC, particulate organic carbon, from satellite observations. Some of them use a band ratio similar to the chlorophyll algorithms we showed you previously. Others use these semi-analytical approaches to get the backscattering coefficient and then relate the backscattering to POC. And this is an example of a map of particulate organic carbon in the oceans, from this Haley-Evers-King study in 2017. Now, owing to the large POC data sets we have in the ocean, it's been quite a well-validated product now, and actually the validation has suggested that we can retrieve particulate organic carbon in the ocean with a similar accuracy and precision to our chlorophyll A retrievals. So it's one of the carbon products we're a little bit more confident in from space. Okay, next I'm going to talk about phytoplankton groups. So in the context of the ocean carbon cycle, but also various other biogeochemical cycles in the ocean, the community structure of the phytoplankton plays a pivotal role. Now, in particular, the size of the phytoplankton has been referred to as a master trait, and is certainly important in the context of the ocean carbon cycle. And this image here shows different sizes of phytoplankton increasing from a prochlorococcus through to our diatoms. Now, the size of the cell influences the phytoplankton physiology. It influences their metabolic rates, how much nutrients they can take up. Small cells have a higher surface to volume ratio, so are capable of absorbing nutrients in very oligotrophic, so low nutrient concentrations, giving them a competitive advantage. Small cells are also found to absorb light with higher efficiency. The size of the cell is also thought to be important in the context of export production, so the amount of carbon that sinks down into the deep ocean, and also their sinking rate, so the rate of sinking of these particles. And of course it has a big influence on the marine food web, so how energy is transferred through to zooplankton and up to higher trophic levels. So the phytoplankton size really matters. Now, there's various ways one can look at phytoplankton community structure from space. There are techniques that people have developed which are detection based, so that they look at subtle optical signatures in the ocean colour to distinguish between different communities and sizes of phytoplankton. And there's extrapolation based techniques which relate the community of a structure of phytoplankton to some other variable we are confident we can retrieve from space, like the chlorophyll concentration. And this is just an example of an extrapolation based based approach. This is one of models that I've worked on and developed. And in this approach, basically, we look at relationships between the chlorophyll concentration in three size classes, pico, nano and micro, and how they vary as a function of the total phytoplankton biomass, the satellite chlorophyll, which we can retrieve from space. So if we develop these conceptual models, in this case, that relate these variables, we can take a satellite chlorophyll field and we can produce maps of our predictions of the different size classes of phytoplankton. Their chlorophyll, ultimately their carbon, which can then, give us a route into some of these ocean carbon cycle processes like export production. Now, initial validations of these products are they're ongoing, but they indicate we can retrieve these different size classes with reasonable accuracy. Some are a little bit more tricky than others, but typically we're looking at errors in the order of 0.3 to 0.4, and that's log 10 chlorophyll. So we often do our validation statistics in log space when we're dealing with chlorophyll, owing to the log normal distribution of chlorophyll in the oceans. Okay, next we have particulate inorganic carbon, so these calcium carbonate pools. Now, this is one of the products that actually has had a long history in satellite ocean colour remote sensing. So this is an example on the top left here of a coccolithophore bloom, actually near where I live, in the southwest of the UK here. Now, coccolithophores produce these calcium carbonate shells called liths, shown in the top right image there. Now, when the cells die, they release these liths, and they can scatter light or backscatter light with very high intensity, such that the signals can be easily seen from space, as shown in the image on the top left there. And scientists have developed algorithms that use the ocean colour observations in order to determine the concentration of particulate inorganic carbon, which is primarily coming from these liths in the phytoplankton here. And this is an example on the bottom left of a map of particulate inorganic carbon and some validation studies here. This paper from Catherine Mitchell in 2017 in the bottom right there. So satellite observations can be used to look at these particulate inorganic carbon pools with pretty good accuracy and then understand their dynamics in the ocean. Now, next we're going to talk about primary production. So primary production is really the engine behind the ocean biological carbon pump. So what is it? Well, it's basically the conversion of inorganic carbon into organic carbon through the process of photosynthesis. Okay. So in order to model primary production or map it from satellite observations, we essentially need four pieces of information. We need to know the biomass of phytoplankton, which we can retrieve using satellite observations of chlorophyll concentration, for example. We need to know the light environment. We can retrieve the photosynthetically available radiation from satellite observations, and that can be used to model the light environment in the ocean with additional knowledge on the diffuse attenuation coefficient, which we can get. So we can get the biomass, we can model the light environment. Now, the two other pieces of information we need are the parameters of the photosynthesis irradiance curve. And that's shown on the top left image here, where we have a production per unit biomass, so normalized production, as a function of light. And this can be described using this classical photosynthesis irradiance equation with two parameters. The maximum photosynthetic rate, denoted here as PBM, and the initial slope of this curve. Now, with those two parameters and our biomass and our light fields, we can produce maps of primary production. But it's the parameters of the photosynthesis irradiance curve that's really tricky to get at. And techniques are being developed to predict these parameters from satellite observations, as shown in this image here for the PV max. Now, with better parameters, we can then model our primary production with higher accuracy. And this is just an example of a primary production map in the North Atlantic in August 2003. And there are various techniques that have been developed to model primary production. Now, in terms of validation, this is just a study from Saba in 2011, illustrating kind of the levels of accuracy that these satellite primary production models have. Again, we're looking at a deviation or root mean square area difference with in situ observations of around 0.3 to 0.4 in log space. Okay, so that's our primary production. We can also try to derive maps of export production. And various techniques are being developed to do this at present. The technique on the left here is an empirical algorithm that's been developed by Laws et al. In 2011, and there are other algorithms that have looked into this. And they relate the they firstly map the export efficiency using this empirical equation at the top here, using two variables we can retrieve from satellite: temperature fields and total primary production, which I showed you in the previous slide. They then estimate the export efficiency. They multiply that by the primary production in order to estimate how much of the primary production has been exported to the interior ocean. And as I stated at the beginning, this is estimated at around 9 to 12 gigatons of carbon in the ocean. Now, there is a lot of work going on as well, certainly by through the NASA Exports program, that are trying to develop more mechanistic algorithms that can improve the export production rates. So moving away from empirical algorithms towards more mechanistic, taking information about the community structure of the phytoplankton, the small and large cells, information from the zooplankton and primary production, and funnel that through a mechanistic model in order to improve export production estimates. Now, in terms of the dissolved organic carbon pool, we can access some information on dissolved organic carbon through the satellite colour dissolved organic matter product. And this is particularly true in coastal regions. So in this slide here, on the top left here, we have the optical properties of water, pigments and Gelbstoff or CDOM, which is the brown line here. And you can see it has this exponential decrease in absorption with increasing wavelength. And there are semi-analytical algorithms that can retrieve this absorption coefficient using satellite observations. Now, there's a lot of work being done in coastal regions and also in the Arctic regions as well, that have looked at the relationship between the colour dissolved organic matter absorption and the dissolved organic carbon concentration in the ocean, and finding very tight relationships in some cases in the Arctic, some nice relationships in the northeast coast of the United States as well, showing that these relationships do vary a little bit depending on the season. And with the CDOM absorption maps, one can then convert them into DOC concentrations and look at the distribution of DOC in the coastal ocean. However, the difficulty in using this really comes to play when we look at the open ocean. Now, work by Nelson and Siegel in 2013 really demonstrated that there is not a strong relationship between the DOC concentration and the CDOM absorption in the open ocean. Whereas we saw tight relationships around the coastal regions, often related to a river runoff and high CDOM concentrations correlating with high DOC concentrations. In the open ocean, that's not the case. And so it's a real challenging product to retrieve using satellite observations in the open ocean. And some of the work we're doing in BICEP is looking to improve satellite models, introducing additional information on temperature, salinity, and other variables we can retrieve from space in order to try to improve maps of DOC concentration. And finally, the product I'm going to look at a little bit now is the is the PCO2 concentration in water. So the exchange of CO2 between the ocean and the atmosphere depends on the gradient in CO2 between the ocean and the atmosphere. When the atmosphere has higher CO2 concentrations than the ocean, then there's going to be a gradient from the atmosphere to the ocean, and vice versa. When the ocean CO2 concentrations are higher than the atmosphere, there's going to be a release of CO2 from the ocean to the atmosphere. And that air-sea flux also depends on a whole range of other factors, temperature and wind speed, and other factors are related to the interface between the ocean and the atmosphere. Techniques have been developed to estimate the particle pressure of CO2 in the ocean, and these techniques typically involve developing statistical relationships between PCO2 and variables we can retrieve from space. So this is just an example from the ONO paper in 2004 that estimated PCO2 as a function of temperature and chlorophyll. Now, one thing to bear in mind with satellite observations is there are limitations. I mean, we discussed this a little bit early on with the fact that satellites can't see through clouds. So in cloudy conditions, or in locations of the planet that suffer from high cloud coverage, it's going to be difficult to do. There are also passive sensors we use, so they can't see the polar regions in the winter, though increasingly now LiDAR satellite remote sensing is starting to fill those gaps. But one of the biggest limitations in terms of the ocean carbon cycle is that it can only see the surface layer of the ocean. So, and as I discussed earlier, typically in the open ocean this is around 10 meters, can be as deep as 40 meters in the clearest waters. Now, the euphotic depth, where all the primary production happens, where there's enough light for photosynthesis, can be as deep as 150 meters in some regions. So extrapolating the surface observations we see from satellite to depth is challenging. Now, fortunately for us, there are new tools that are being used in oceanography, robotic platforms, and one of these tools is Argo floats. So this is an example of a biogeochemical Argo float, which in addition to measuring physical salinity and temperature, also measures biogeochemical variables, measures the chlorophyll concentration and the backscattering, which we can link with our satellite observations. Now, these floats are very clever things. You put them into the ocean, they sink to around 1,000 meters, and then once a week they drop down to 2,000 meters, do a vertical profile of the ocean, collecting chlorophyll data, temperature, salinity and other variables, and then send that data via satellite. So there's a lot of science now going on where they integrate data from these floats with satellite observations in order to paint a better picture of processes related to the ocean carbon cycle. And this is just one example here from a paper from a colleague who looked at fluctuations in carbon to depth driven by what they called the mixed layer pumps. So fluctuations in the mixed layer before the spring bloom was pumping lots of carbon down to depth. So this is the real frontier in ocean carbon monitoring at the moment, linking satellite observations with these robotic platforms. And we can also link our satellite observations with models. So as we showed in that slide a bit earlier on, there are many pools and fluxes of carbon that we can't see from space. One thing we can do is we can develop mathematical models of our ocean that look at these pools and fluxes, and we can drive them, we can assimilate data into them in order to improve our understanding of the fluxes and the pools we can't see from satellites. Okay, so that's part 2 of the lecture. So we've looked at the Earth's carbon cycle, and the application we're looking at here is the use of ocean colour observations for understanding the marine carbon cycle, the ocean biological carbon pump. We've looked at various satellite products that do this, POC, PIC, and the DOC related products. The importance of integrating these with robotic platforms and with models.